More than 150 years ago, Schleiden and Schwann proposed that all living things are made of cells, that cells are the basic units of life, and that cells come from pre-existing cells. Let's focus on the second point of the cell theory. Cells are the basic units of life. As we saw earlier, cells function as independent biological factories, each with the equipment, assembly lines, and transportation systems to make whatever it needs for energy or to repair its structures. Many of a cell's functions are carried out by organelles, distinct structures or compartments within a eukaryotic cell that perform specific tasks. We'll learn more about cells as we construct a model of a human liver cell in this activity. Take some time to review what we've learned about cell structure and function. See if you can identify the structures and organelles. Quiz yourself, then roll your pointer over the structure to see its name. Click on an organelle or molecule to review its structure and function. The cytosol is the fluid part of the cell, excluding the organelles. It's an aqueous mixture containing ions, small molecules, and soluble macromolecules. Smooth ER is part of a network of membranes adjacent to the nucleus. Its primary function is to synthesize and break down small molecules. Rough ER is part of a network of membranes adjacent to the nucleus. It contains ribosomes that synthesize proteins destined to be packaged inside organelles or released outside the cell. The extracellular matrix is a complex mixture of proteins and carbohydrates in which cells are embedded. Click More Detail to review the structure of the extracellular matrix. Proteins called integrins span the plasma membrane and interact with fibronectin on the extracellular side. The fibronectin molecules associate with collagen fibers outside the cell. Finally, the collagen fibers are anchored in place by proteoglycans, large molecules containing both protein and carbohydrate. The Golgi apparatus is a membranous organelle that plays a critical role in modifying, packaging, and routing proteins produced in the endoplasmic reticulum. Mitochondria are the cell's main energy factories. They convert the chemical energy in food to a more usable form, adenosine triphosphate, or ATP. Click More Detail to review the structure of the mitochondrion. The mitochondrion has a double membrane, but the inner membrane contains a complex series of folds called cristae. The chemical reactions involved in converting food energy to ATP take place on these folds, as well as in the mitochondrial matrix. Lysosomes are organelles involved in the breakdown of biological molecules. They contain a variety of digestive enzymes that function best in the acidic environment of the lysosome, but are relatively inactive at the neutral pH of the cytosol. The nucleus is a membrane-enclosed organelle containing most of a cell's genetic material. Click More Detail to review the structure of the nucleus. The nucleus is the largest organelle in eukaryotic cells. It contains a double membrane covered with pores. The interior is a mixture of genetic material called chromatin, suspended in nucleoplasm. There's also a nucleolus, which synthesizes the components of ribosomes. Peroxisomes are membrane-enclosed organelles responsible for the breakdown of toxic substances. The ribosome is the site of protein synthesis. Ribosomes can float freely in the cytosol, or they can be bound to the endoplasmic reticulum. The cytoskeleton is a three-dimensional network of microtubules and filaments. It provides internal support for the cells, anchors internal cell structures, and participates in cell movement and division.